Vegetal proteins in enology is the title. Well, thank you, Gianni, for your introduction. Good afternoon. This module focuses on vegetal proteins, and I will tell you what they are, how they are, and what they do in the winemaking applications. 4 points in the agenda. A very brief historic introduction. Then I will tell you about the coming back of vegetal proteins, and I will frame that in a context. I will explain to you why I'm talking about coming back. Then I will talk about sources, which is the processes by which vegetal proteins are produced, and then their features, their chemical features. Then I will make a number of examples of applications that testify to their behavior in winemaking. History. Let's go back 2,000 years. Why not? I would like to briefly hint at what happened among the Romans 2,000 years ago. And why am I talking about the Romans? They did not invent enology. They received the foundations of winemaking from the Etruscans, who in turn received that from the Greeks. And in the morning, Professor Moyo went back to the Neolithic age, but I will stop with the Romans. Romans did something special in that they made wine a daily beverage, and they disseminated the culture of wine. Wine became part of the life, ordinary life of people, not limited to an elite group. Nutrition was also an extra benefit coming from wine at the time, and entering people's daily lives also entailed that higher volumes were produced, which entailed the management capabilities that were required. Various documents ascertain that production volumes were not excellent. And here I again make reference to Professor Moyo's presentation. He talked about the addition of aromas. That was a common thing. But not only that, wine was prepared so that it could be presented in different ways. It would be mixed with other quantities or blended, and it was fined as well. Fining was common, and fining was performed with what was available, meaning that the ancestors of winemaking products already existed at the time. Here you have a number of examples. So milk from both cows and goat milk, eggs, the yolk was used extensively, the white was used extensively, and pea meal, because peas were grown extensively at the time. Rubiglio was very widespread. It's half in between a lentil and a pea, and it is grown still in the area of the Sibillini Mountains. Then meal was made out of those peas, and it was added to wine. And it was because of the proteins that the meal contained. Of course, the Romans did not know about that. They just saw the effect. So you see that proteins of vegetal origin were already used at the time. And this is the reason why I'm talking about a coming back of vegetal proteins in enology. which is something which should be framed in a better context. And now I will come to the present time. You see, I would like to subdivide the period into two halves, and the limit between the two halves are the 90s. Animal proteins were used until the 90s normally for fining purposes. And I think that you all know what they are. We think about albumin, gels with various content, and potassium caseate. So there were criticalities, and criticality, the Creutzfeldt-Jakob disease was one of them. So I'm really using a term, criticality, which is not perfectly correct. It was a huge problem due to the Creutzfeldt-Jakob disease. Animal proteins were seen differently, and that had an impact in terms of considering proteins in general. Also, allergies increased, and new market trends came to the fore. In that, new needs emerged in terms of requests for wines for vegans and vegetarians. All this entailed reduced use of animal proteins to the benefit of vegetal ones. Many companies at the time studied applications of proteins obtained from various sources. I would say that almost all were tested, from leguminous plants, from pulses, and I'm referring to soy proteins, or beans, or peas, or wheat, also cereals, and here I'm referring to gluten, of course, and also potatoes. All these were tested. Their performance in enological terms was tested. Then, due to various parameters, indeed two proteins emerged as being better, which is proteins from peas and from potatoes. And I will focus on them from now on. What about the sources of pea and potato proteins, and what about their chemical features? I will try and be brief in providing you with this information. Production. How do we produce pea proteins? Of course, the seed is the source. It is separated from the skin. Then the meal is produced after crushing it, and what is soluble remains in a solution, and the remaining part is separated from the rest using, for instance, centrifugal forces and methods. What is obtained, and that is the protein in a solution, needs to separate it from the rest. pH is the way to do so, in addition to temperature. By combining the two things, you have a purified protein. The protein is separated, it is atomized, and you have the final concentrated product. Production of potato proteins is different in the initial steps, in that the protein from potatoes is obtained from a process which is parallel to the production of starch or potatoes, which are then used for frying. Imagine that approximately 50 million tons of potatoes are obtained, 40% of them are processed, and a major quantity of processed products are starch on the one hand and potatoes which are fried. Proteins need to be separated from the starch in order for starch to be pure, and the proteins then enter a different processing pathway and they are purified. When you prepare potatoes for frying, then the cut pieces of potatoes are put into water. The starch portion and the protein portion on the surface should be removed to improve the frying outcome. The solution with starch and protein then is purified, and you see that the phases are exactly the same, working on the isoelectric point, and then atomization and concentrated product. So I think that this is an interesting process. Purification is the core of the production chain for both products, for both proteins. Now, the chemistry. Pea proteins are what you see there: legumin, viscellin, conviscellin. They are globulins. They can be aggregated in fairly complex forms. Their molecular mass ranges from 380 to 150 kilodalton, and the isoelectric point ranges from 4.5 to 5.8, which is very close to the potatoes. In that respect, while all the rest is different, it is glycoproteins. Their form is very different, and they have more linear portions and also helical parts. The molecular portion is smaller, 40 kilodaltons. What about their presence in enology? In enology, and this is a key message, they are suspended. The percentage of solubility in function of pH, this is what is represented there. You notice a red rectangular shape. You see that solubility of pea proteins is approximately 15%, which is low, but the potato protein has even lower volumes, which means that they are in a suspension, these proteins. In order to evidence what that means, I show to you this picture. This is their state. On the left-hand side, you see peas, and on the right-hand side, the potato proteins. So it would be nice to see how the microparticles of the protein aggregates look like in a suspension in the pH. And you can actually have a view of them with electron microscopes. You can have a picture. These are images that were captured after magnifying 3,000 times. On the left-hand side, you see the protein aggregate of pea proteins. On the right, you have the potato proteins. They look different, and I might describe the former as deflated, in a way, peas. And this is what you find on the market. And then the protein from potatoes look like sponges. The proteins do have an enologic effect. and that is linked to surface interactions. That is the charge on the surface and the hydrophobic feature. A small percentage of these products end up in the solution. You see here the two electrophoretic profiles that you find at the center on the right-hand side. So potato again and pea proteins. You see here the bands that represent the portions of molecular mass which are most present in the solution. This is the prevailing band for the potato protein, and we talk about approximately 40 kilodaltons as a mass, while we have several bands for the pea protein, which means that we'll have a highly distributed series of bands, which means that, again, we have a difference between the two. Let's now focus on their enological behavior. What about their performance when applying them? What can we expect from them? I started from the easiest thing. So the fact that they fine the product. Do they always have a fining activity? Well, they do, correct. They make the wine less hazy, that is for sure, and their effectiveness in this regard can be seen here in the two colors. So they have a clarifying effect. And the lower the bars, the better the effect, because then you have less hazy wine. And it seems that peas do better here. Now, we have two different wines, Trebbiano and Sagrantino. If the proteins are properly processed, then they can perform even better, which is what is shown here after applying a moderate hydrolysis to the potato and pea proteins. What does that mean? Well, you need to get to a hydrolysis point which gives the best performance without going beyond that, because that would trigger a negative effect. Instead, if we make an average in absolute terms, pea proteins have the best performance. That is our winner. While potato proteins with a moderate hydrolysis works nicely. I don't want to diminish the importance of the clarification effect. Here you see two tests on Sauvignon Blanc and Trebbiano, which was also included in the previous graph. In the right-hand cylinder or tube, you see that evidently you have a less hazy wine, and there moderate hydrolysis was applied to potato proteins. So there is an effect also in potato proteins and hydrolysis. Now, the clarifying activity is also important when it comes to the flotation aspect and portion, and you see the various groups. This is a flotation test performed with a fairly simple juice starting from 600 NTU, and I believe though that the general behavior is important. Here we can confirm again that pea protein is the protein which best fits this application. I hope you can see the difference here because these images actually testify to the fact that the numbers I gave you are correct. There we have a test from flotation, and it is a pea protein moderate hydrolysis. It is fairly clear. Also in terms of color, I would say that it is the best test, very similar to the picture on the right, which is where we used the swine gel. And I think that you all know about the performance of gel in flotation. That was taken as a reference. Now, here we have data about oxidation management, because vegetal proteins are also asked that to what extent can they manage oxidation. Indeed, here we see optical density 420 nanometers for wines, and the performance of the two proteins slightly differs. The best effect in terms of color is obtained with pea proteins. If you calculate the average, that is the outcome. And that goes for immediate clarification. And then pea proteins also work best in preventing oxidation if you consider their capability of extracting iron. Here on the right-hand hand, you have the average reduction of the iron percentage. The higher the bar, the better the result. Proteins from peas extract or eliminate more iron than potatoes. And the reference is PVI PVP, which is the best product to take away iron. So again, pea protein is the best. Now, astringency. What about proteins that can limit astringency in wines? 3 different wines were tested with several proteins taken from the marketed products. If you compare and contrast the height of the bars in gray, yellow, and orange, and compare and contrast them with the blue, which is the control, you see that the gray bar is about potato protein, and it is the best one. Best performance in terms of reducing astringency. Now, very briefly, I would like to mention the sensory profile impact. I did not want to dwell upon the density of the profile. It would take so long, but I will simply say that if you need to clarify and find wines, then if you use these compounds, you can improve the sensory profile very much. And the olfactory notes are cleaner. Vegetal note is something I should mention, in that vegetal proteins have unintended vegetal notes with them. This was very true until a few years ago. Now this problem has basically been solved. The products that are proposed are selected in terms of improving that, and so selection is important. It is very important to use the right dosage and to also make application tests. This is no longer an issue. That is the point. It is not something that we need to focus our attention on very much in order to improve it. Conclusion. I would like to underline 2 points to conclude with, and they are my take-home messages. First, for sure, pea and potato proteins have specific applications in winemaking, and that has been evidenced. Second, these proteins can be produced with tailored features for winemaking applications, which means that their application can be extended, and we have seen examples of that. Now, I really conclude, and I do so by thanking a number of people working in the research team of Enartis that in Italy is responsible for the tests that I showed to you. And I thank you very much for your attention.
Presento io allora. Mi tocca guadagnarmi lo stipendio da moderatore.
Okay. Now we have Matteo Marangon of the University of Padova. I think he's well known for his studies on macromolecules and especially terpenes in wine. He will discuss today the plant proteins as a tool to manage colloids. You can ask your questions on Slido as usual, and if there's time at the end of the presentations, we'll have time to take some of those questions. Thank you, Gianni. Thanks, everybody, for being here. As you said in your introduction, I deal with proteins in wine, and plant proteins are proteins. Let me explain the agenda. I will start by devoting some attention to colloids in reds and whites and the properties of these substances, of these compounds. We will mention the key aspects of colloidal instability in wines and how they can and how such instability can be managed. And then I will correlate that with the possible use of plant proteins in the management of wine colloids. Now, colloids. If you followed some of our webinars, you may have seen this slide already. Colloids are compounds that if you use from a size perspective, you find soluble compounds, alcohols, organic acids, sugars, and monomeric polyphenols. There's a class then of medium-sized compounds from 1 nanometer to 1 micron, defined as colloidal compounds, commonly the macromolecules, proteins, polysaccharides, and condensed polyphenols. They are not large enough to be visible and therefore to cause turbidity, in other words, haziness or cloudiness, but they are potentially unstable and therefore they can give rise to certain problems under certain conditions, or they can be potentially stable and they can remain as stable colloids without settling on the bottom. Enologists know well that this can give rise to aggregations if they're unstable, when they're unstable, that will settle over time. Not every filtration can eliminate them, but ultrafiltrations, for instance, can remove them, thus changing the colloidal status of wines. And then you have the compounds that are visible to the naked eye, cells, crystals, and large aggregates. These are easy to see and can be easily removed by filtration. Some characteristics that it is useful to know to understand how colloids behave in wines. Colloids are not still in wines, but they move around. This is what the video is showing. You see the colloids and the fact that they follow a certain movement pattern driven by Brownian motion. So they will continue to float in the wine all the time. Given their size, they can clog the filters, they can flocculate also regardless of their concentration. They can grow and become insoluble over time. They can refract and disperse light, and therefore they can be perceived as turbidity. They are electrically charged with surface charges that gives them the property to repulse other objects. But when the charge is lost during aging, storage, and other conditions, the interactions between molecules that are no longer charged may no longer exist, and therefore they can aggregate, grow in size, and flocculate. In terms of the types of colloids, there are essentially two types of colloids: the condensing or condensed polyphenols formed by the micellar or associative colloids that are linked by low-level bonds, and over time they can cause destabilization, they can flocculate or precipitate and cause turbidity. But then you also have macromolecular colloids, polysaccharides or proteins bound by covalent bonds, hydrophilic, soluble in water, and easy to hydrate. And this, alongside the fact that they are charged, makes them more stable because they are hydrophilic and charged, and therefore their interaction with similar colloids is prevented or delayed. An important fact is that macromolecular colloids can stabilize the association colloids, making them more stable, while naturally they're not stable, by coating them and protecting them, making them more easily hydratable, with forming an additional protective layer. I was discussing macromolecules in wine. The compounds that contribute to the formation of colloids are proteins, generally. There are more proteins in white wines because they are not treated using bentonite than in reds, and then total polysaccharides that can vary depending on the type of wine, and that is also impacted by maceration or lack thereof. Then total polyphenols that can be not be extracted in white wines unless you take maceration, and then total colloids with low proportions and proportions as high as 1.5 grams in red wines. Now that we've understood the properties of colloids, let's see what issues they may cause. In white wines, the protein Cas, but also color precipitation, polyphenol precipitation, quercetin precipitation, and we've heard this morning that Professor Moyo was referring to these potential problems, and that regardless of the type of colloidal instability. This is ascribable essentially to three phenomena, the first being the change in compound solubility. Polyphenols, proteins, the colloidal compounds that are stabilized, and that depends on the chemical activity and its interactions with other molecules, denaturation or co-precipitation in bond with other molecules. There can also be chemical physical interactions with other colloidal compounds, different colloids which, losing their charge, will interact among them, grow and destabilize, or chemical physical and chemical interactions with adjuvants that are added to destabilize the colloids and remove them, so it's an induced and intentional instability. How do you manage such instability? From a protein precipitation perspective, the most frequently used methodology is bentonite, but flash pasteurization, enzyme-based treatments, etc., are also a possibility, although bentonite is the reference methodology. For tartaric instability, cold stabilization, membrane bladder processes, potassium polyaspartate, CMC, and monoproteins to keep the tartrate crystals low. Quercetin precipitation via the remotion using PVPP, charcoal oxygenation during storage, and color instability, where you favor co-pigmentation to prevent it, or you can add protective colloids such as gum arabic and monoproteins, or the removal of unstable color using bentonite and animal proteins, especially gelatin, which is charged. Solubility of phenolic compounds. This is a subclass of instabilities. It can change over time. That's a well-known factor to all those who make wine, because polyphenols, also because of microoxygenation taking place in our tanks and vats, induces self-oxidation, and this causes polyphenols to be more active, to form aggregates that may lead to precipitates. Oxidized tannins, when you see them at this stage, can become reactive, grow over time, bind others, and form precipitates, as shown at the top of the diagram. It also may be that plant or animal proteins can be added for fining, in the form of glue fining, to make these precipitates bigger, make them insoluble, and more easily remove them. Alternatively, you can keep them in a solution through the addition of protective colloids, that is, polysaccharides that can bind and cover the unstable colloid system, making it more stable because of the hydration they will impart to the colloids, as well as the charge that they will transfer. Now, this all was necessary before we move on to data on plant proteins and the effect that these plant proteins can have on colloid stability and the necessary consequences thereof. As we heard from Gianni, plant proteins can be used for a number of reasons, to fine, to manage oxidized polyphenols, and to moderate the astringency in wine. But the question that one could ask from a scientific point of view is, what effects would the addition of plant proteins have on the colloids in white wines and potentially red wines as well? Now, what we know, also based on what we've just heard, is that plant proteins from peas and potatoes are a lot less soluble. Soluble with respect to the wine's pH than animal proteins. This characteristic, as Gianni showed, is associated to the production process that may result in less soluble compounds that favor hydrophobic interactions, because the hydrophobic binding sites are more exposed, and they can be more effective when they are added to the wines in the binding of molecules. Their surface charge is positive at the wine's pH. Generally, the value of P is 4 to 6, and what changes a lot compared to the products commonly sold is the different degrees of hydrolysis. The more they are hydrolyzed, the more the contact and surface and solubility will increase, so to respond to the intended applications. Here are data from our labs. This is the total protein content in a Traminer wine. Red is the control samples. We're talking about a wine with 85 liters of milligrams of proteins per liter of wine, and then you have the samples with pea proteins 1, 2, 3 and hydrolyzed protein, and then potatoes 1, 2, 3 and 4 for hydrolyzed, and then potato and pea mixed plant proteins, caseinate, non-hydrolyzed gelatin, hydrolyzed gelatin and fish glue or isinglass. In 7 cases out of 12, plant proteins induced minor reductions in protein content. The maximum is minus 11% in the case of this product containing potato protein. And this is more often the tomatin-like proteins that are reduced. And then in some cases you even have an increase due to the fact that part of the proteins that are affected are soluble. But we're talking about very moderate changes versus the total protein content. The next step is to see if the ones are more or less stable to heat. In this case, 80 degrees for 30 minutes. The control is always the untreated wine in red. This traminer had an instability equal to 50 NTUs, so heat instability that was relatively high. If you look at the impacts, it's very diverse. There are products like these where there's a reduction in cloudiness by 20% after the heat test, and others where it's plus 20% instead of minus 20%. So it's harder to relate the total protein content with the data of on instability in this case. So there's no strong relationship between the total protein content and the heat instability. They're necessary, therefore there have to be other elements that play a role in this. Okay, the biggest increases are seen in products that are derived from potatoes, where the variations were almost irrelevant from a protein perspective. There might have been an interaction with other wine compounds that can lead to this instability. In each and every case where a reduction in instability was seen, 20%, 4, 8, 10%, there are always pea proteins involved. So apparently these proteins derived from peas may play a role. Another wine that is less rich in proteins than the previous one is Trebbiano. The content in this case around 24 milligrams per liter in total. The green bars were produced by the various pea proteins. The reduction levels were not high, but higher than the previous wine. The percentages are more significant than the previous wine. The matrix is different, and hence the interaction of protein extracts and the proteins in wine is different, and so that amplifies as well the removal proportion. With pea proteins, reductions go from -4 to -17% in concentration, and in this case again it's most effective on tomatins. Look now at the heat test. The wine wasn't very unstable at the beginning. We're talking about 3.5 NTUs, so not a very unstable wine. In any case, with all the treatments carried out, the wines lost a good proportion of their instability. I didn't indicate the percentage proportions. They wouldn't have been high, 2-3%. But in many of these cases, the wine would become more stable after treatment. An additional analysis we carried out was to take the protein extracts and we analyzed them. We used a methodology to measure the polysaccharide content, weight for weight in milligrams per kilo. And we wanted to show that these protein extracts, as per the OIV resolution, needs to have a minimum content of 65% a minimum a protein content of 65%. And that is met, but there's a great heterogeneity in polysaccharides among these different colloids. And polysaccharides were almost exclusively low molecular weight. Now, the question we had asked initially was why are unstable wines treated with plant proteins more stable to heat despite their moderate reduction in proteins? Well, that would appear to be not logical at all. So we formulated a few assumptions to see, based on the structure of these extracts and proteins, what the cause might be, what mechanisms might be at play. The first assumption is that there's an indirect mechanism. It is possible that based on the color of wine and the characteristics of the vegetal proteins, that the polyphenols that have a covalent bond to proteins may indirectly be removed. Hence, the proteins in wine are removed because they are attached, bonded to the polyphenols, and that might be one mechanism. A second indirect mechanism could be that there is a possible inhibition of the tannin to protein bond. We've just talked about the covalent bonds, and it's possible that these extracts may impede this bond, resulting in less aggregation. Between the two assumptions, I tend to prefer assumption number 1, but I cannot rule out assumption number 2. A third assumption is centered around a direct mechanism. These proteins, these preparations are highly insoluble, and therefore there might be an adsorption of these proteins by the insoluble protein extracts, thus a hydrophobic interaction. And then a second assumption around a direct mechanism is that these proteins would bind to reactive polyphenols, causing precipitation that would involve some associated proteins associated to the reactive polyphenols. These are all theoretical assumptions that we'll have to test. Some appear to be more likely than others, but looking at the structure and characteristics of the colloids, these should be the main assumptions that we should test. Okay, my last slide with some final conclusions. I've showed to you some data that proved that plant proteins would lead to moderate removal of proteins, at least in white wines, that the removal mechanisms could be a direct interaction with reactive polyphenols and or indirect with proteins and or protein polyphenol covalent complexes, leading to a reduction in polyphenols in wine, slowing down the colloidal aggregation processes. Plant proteins are potentially also helpful This should be tested again, but they could be potentially helpful in improving colloidal stability in that they contribute to reducing unstable molecules, polyphenols, and perhaps proteins as well, directly or indirectly, at least in white wines. But I think it would be interesting to understand their behavior in red wines as well. And also, it should be mentioned that the mechanisms of these interactions should be better understood with better targeted studies. For instance, using the degree of hydrolysis, the surface charge, the degree of hydrophobia and hydrophilia in preparations. And that concludes my presentation. I would like to thank the Enartis team that works at our lab. I see them all seated in the audience, and all of you for listening to me. Thank you very much.